So from our previous lesson, we were able to describe glycolysis, where we saw that it is a stage where the glucose molecule is successfully converted into pyruvic acid. Okay, and it is divided into two stages: the energy investing stage, okay, where you invest the two ATP molecules to form a high energy fructose 1,6-bisphosphate, which will split to give two 3-6 compounds, that is glyceraldehyde, 3-phosphate, and dihydroxyacetone phosphate. Then each, we say that they are interconvertible, okay, and so since they are interconvertible, then they will proceed, okay, but we can't follow the two, we follow one since they are interconvertible. But whatever is happening on this one, it is also happening on the other one. So where we saw that our PGAL, phosphoglyceraldehyde, it undergoes oxidative phosphorylation, where it loses the reduced NAD, the hydrogen ions, and they are picked up by the NAD to form our 1,3-bisphosphoglycerate. And then you invest another ATP molecule, so that you lose an ATP, you can see, by substrate level phosphorylation, you form 3-phosphoglycerate, and then you isomerize, okay, you transfer a phosphor group from carbon 3 to carbon 2, okay, and then after that you lose a water molecule. Finally you lose an ATP molecule to form pyruvic acid. Now pyruvic acid, it is a product of glycolysis. It is formed in glycolysis. Now the next is, what are the products of glycolysis, or what is the significance of glycolysis as a process? So number one, glycolysis, let me get some pen here. It's going to give you your ATP, okay, because we have seen that we invest two ATP molecules. You invest two, you produce 4, and then after 4 minus 2, the one you've used, so you produce a net of 2 ATP molecules. So for these organisms that do undergo anaerobic respiration, that's how they just produce 2 ATP molecules. However, it's robust, okay, it's rapid. However, you produce just 2 ATP molecules. Then it gives us, the glycolysis gives us the NAD and the pyruvic acid that proceed to further stages in presence of oxygen, okay. But if oxygen is absent, then they undergo either alcoholic fermentation or lactic acid fermentation, okay, where the pyruvic acid is converted into an alcohol or lactic acid, depending on the compound that is undergoing that anaerobic respiration, okay. And these products are important, these intermediate steps. If glycolysis is not there, then there is no way how the other processes will occur, okay. There is no way how. You cannot have Krebs cycle, neither can you have the electron transport system, okay. And so you must first undergo glycolysis to produce the pyruvic acid, that in presence of oxygen, then the pyruvic acid is going to be shunned into the mitochondrion for further oxidation, okay. And the NAD, the reduced NAD, is also shunned to the membrane system for further oxidation to produce the energy, okay. Yes. But that happens only if oxygen is there. But in absence of oxygen, in absence of molecular oxygen, then both the NAD, the reduced NAD, and the pyruvic acid, they undergo fermentation, okay, to regenerate back the NAD, the oxidized NAD. And then the pyruvic acid is either converted into an alcohol or lactic acid, depending on the organism that is undergoing that type of respiration. So we have a note or NB. Glycolysis, it yields about 5.2% of the ATP produced in the body cells. Imagine if you produce 38 ATP molecules, and with glycolysis you produce an 80 of 2 ATP molecules. So if you get your 2 out of the total 38 that is produced by the entire respiration, then times 100, you should be in position to come up with the 5.2%. And we are going to account for ATP, by the way, so just be calm, okay? Have no worry, just be calm. Now this is the question that I asked you the other time, and no guy responded. All of you are like, man, we have no idea. All of you are like, man, we can't do this question. But it is simple. Basing on the glycolysis, on what you have studied, on the information that you have about glycolysis, how can you use it as a basis, okay, of evolution? And it is simple, it is so interesting, okay? If you've ever heard of the endosymbiotic theory, okay? The endosymbiotic theory, if you've ever heard of it, then you can agree with me, okay? Basing on these evidences. Number one, the ancient prokaryotes probably used glycolysis to make ATP. Why? Because the ancient atmosphere was oxygen deficient. Okay? If you do carbon dating and you date to around 2 billion years, I think I have the year here. Yeah, around 3.5 billion years ago, the environment had little or no oxygen. Why? Because we didn't have the photosynthetic organisms, the plants and the bacteria. They were not there. So therefore oxygen was not abundant like in today's atmosphere. Okay? But due to emergence of these photosynthetic organisms, the bacteria, the photosynthetic bacteria, which later evolved into plants, then today's atmosphere is rich in oxygen. Okay? So as a result, these ancient bacteria, which were first to evolve in such an environment where no oxygen was there, then probably they depended on glycolysis, only glycolysis, to produce their energy. Because the glycolysis, it does not, or it just occurs in the cytoplasm. And so it doesn't need oxygen to occur. Whether oxygen is available, whether oxygen is not available, for it will proceed. Okay? Yeah. So that's why when you start sprinting right now, and you can no longer supply enough oxygen, then you resort to glycolysis. It just takes place within the cytoplasm of a cell, and it doesn't need oxygen to take place. So this one, it's a good evolutionary significance to try to explain that really evolution took place. First of all, why is it universal to all organisms? Why is it that all organisms, they do carry out glycolysis? All of them, right from the archaea up to us, the most advanced organisms. Why is it that they all do carry out glycolysis? And actually, glycolysis occurs from where? In eukaryotic cells. Glycolysis occurs within the cytoplasm. Okay? But now the rest, so why? So probably that becomes a reason. So oxygen, it accumulated by this time. Okay? And so from that, these earlier prokaryotes probably may have generated ATP exclusively from glycolysis, which does not require oxygen. Then it is universal. It occurs amongst all organisms. Okay? And it suggests that probably, why? Why is it that all organisms, the first descendant, the very first organisms to carry out glycolysis, they are the ones that now evolved into all living organisms. Okay? And glycolysis remains. It is located in the cytoplasm where no membranes, okay, are required in eukaryotic cells. For instance, look at this. If you take the mitochondria, for instance, and you study its cell membrane, it is identical, okay? It is identical to the inner membrane. Did I say mitochondria? And if you take, for instance, a bacterium and you study its cell membrane, the membrane is identical to the inner membrane of the mitochondria. And also in bacterial cells, glycolysis, so the electron transport system, occurs within the infoldings of the membrane called mesosome. Even here, in the mitochondrion, the electron transport system occurs where? In the membrane still, in the inner membrane. In those cristae, they are the ones that do carry out electron transport system. So probably, it shows some evolutionary relationship. So now, because these early prokaryotic cells didn't have this, hadn't evolved the mitochondria, okay? You can read about the endosymbiotic theory and see how the mitochondrion and the chloroplasts, how they were evolved into the eukaryotic cells. Probably they depended entirely, okay, on glycolysis. And they had nothing like Krebs cycle, nothing like electron transport system. Why? Because they didn't have the mitochondria. And why? Because the environment was poor in oxygen. So the oxygen, these organelles, they evolved approximately 1 billion years ago, okay, after the prokaryotic cells. So now, basing on all that evidence, we can write an essay to support that glycolysis, or to support the theory of evolution, that evolution really occurs. It occurred. We all came from bacteria cells. All we were once bacteria cells. Not so? But due to some differences in the environments, then we become kinda more complex than just being bacterial cells. Okay? That's pretty good. So now, as we have seen, in respiration, we are forming some NAD, reduced NAD, I was almost saying NADP, NAD, and we shall also find other electron carrier molecules, such as FAD, and so on, FMN. Now, with respiration, it involves more of electrons, and they are these electrons that are moved from one area to another area to another area. And in the process, energy will be given out, which the energy will be used to form our ATP. So you need to know these basic electron carrier molecules, okay, and how they work. What are they? Structures, you don't need the structures because no one is going to tell you to describe the structure of NAD. To describe the structure of, nobody is going to tell. So the first carrier molecule we have NAD, and you are going to encounter it, okay? Actually, you have already encountered it in glycolysis, and it's nicotinamide adenine dinucleotide, okay? So this one is also a nucleic acid, but this one is a dinucleotide, okay? It has two nucleotide molecules. If you have studied nucleic acids, you know what that means. So this one, you can check on it. You have this one nucleotide where you have a ribose sugar, okay, and an organic base, adenine, and then a phosphate group. Then this is another phosphate group, a ribose sugar, and a base. And in this case, the base is nicotinamide, okay? And now it is this base that has the active center, okay? And it's this nitrogen that will accept the electron and release the electron to form those two different forms, okay? The oxidized form and the reduced form. So you must have these electron acceptors, these electron carrier molecules, okay, to be the one to transport these electrons that are at relatively high energy, okay? Oh. It's okay, I'll pick later. I'll call them later. It's okay. So that is your NAD. Then you have another one, the flavin, the FAD, or you can name it as flavin adenine dinucleotide. Now this is the vitamin, the derivative from vitamin, okay? And you can see this is a molecule, okay, that has hydrogen, so it is oxidized into this, and the FAD is reduced. However, nobody is going to task you, okay, to describe the structures of this, but you need to have a hint, okay? You need to have a hint. So this is your dinucleotide, okay? So this is your adenine, the ribose sugar, the same way. You have adenine, the ribose sugar, and then you have two phosphate groups, okay? And the phosphate groups are joined to a flavin molecule. This is a derivative from, I think it's vitamin A, okay? So however, nobody is going to task you to describe all this. So it is the one that receives an electron. So in this format, okay, it is going to be in a reduced form, okay, but it will receive that electron. Now this one can accommodate a maximum of 2 protons, where it will receive this and this, okay. So this is going to be the reduced form, where you can see there is hydrogen here and there is another hydrogen here. So it is the one which accepts an electron together with a proton, okay. Remember, proton is just positively charged and an electron, so you receive both the electron and the proton, and remember they will be possessing relatively high energy. So you receive them and carry them to the electron transport system, and you shunt them, okay, into, and they undergo a series of redox reactions at decreasing energies, where they will lose the energy that they are possessing, and that energy is used to form ATP, as we are going to see by chemiosimosis, as we are going to see, okay. Then you may have FMN. This is the same as FAD, but now this one, it is mononucleotide. It has just one nucleotide, okay, instead of having two nucleotide molecules. So basically it is as simple as this. Let's do the cleaning. So these are the electron carrier molecules that we shall encounter, okay, in our respiration, in our discussion, okay, as far as respiration is concerned. So after glycolysis, obviously, the products of glycolysis that are formed, that is the reduced NAD, okay, and the pyruvic acid, pyruvate, they cannot actually stay in a cell because this reduced NAD is unstable even, and you must regenerate the reduced NAD. So that the oxidized form of NAD, nicotinamide adenine dinucleotide, you must regenerate that form to be able to receive another proton and another electron so that you can maintain, okay, the rate of respiration. That's why you people, even you feel that hangover after you have boozed, after you've taken your OH functional group. In the morning you feel when you have hangover, okay. Now what's the cause of hangover is, I usually say this: ethanol has no nutritious value. You take ethanol, the way you take ethanol is the same way you eliminate it from your body. It does not have any nutritious value. So when you take ethanol, much of it is what we call alcoholic degradation, and it occurs in the liver. So you use reduced NAD, okay, to oxidize the ethanol so that you remove it in the body. And you use much of the NAD, okay, and so in the process you will have more of reduced NADH, okay, and you won't have the oxidized form of NAD. So as a result, since you don't have this oxidized form of NAD, the rate of respiration in your body is going to occur at a slow pace. It will be very low. Why? Because you don't have the NAD. And so if it occurs at a very low rate, then you won't be able to produce the energy. And so you feel that general body weakness. So one of the significance is you must be able to regenerate back the reduced NAD, okay, and you must be able to remove the pyruvic acid, the pyruvate, okay, so that it doesn't alter the pH. This is an acid, pyruvic acid, okay. So if it accumulates, it can alter the pH, it can ionize, alter the pH. So therefore you must find ways, and there are two ways. The first one occurs in presence of oxygen, okay, where the pyruvic acid undergoes oxidative decarboxylation. Is it even oxidative? You add the OH, no, it is reduced. Now depending on the type, if it is anaerobic, okay, in presence of oxygen, let's start with in presence. So in presence of oxygen, the pyruvic acid undergoes what we call a series, okay, of redox reactions, of oxidative decarboxylation reactions, which will lead to converting this pyruvic acid, okay, into, let me say, water, which is a neutral molecule. The electrons within this pyruvic acid, they undergo a series of chemical reactions until when they are converted into water. And you have water energy and carbon dioxide. But now in absence of oxygen, then this pyruvic acid undergoes what we call fermentation. And depending on the animal, if it is a plant and yeast, then it undergoes what we call alcoholic fermentation, where the acid undergoes decarboxylation, okay, and reduction at the same time to form what we call the ethanol, Okay, you decarboxylate, and we are going to study all this. You decarboxylate and then you reduce using the NAD, okay, to form ether. Then here, it's okay, somebody's. A moment, guys, a moment. So, like I was telling you that the fate they undergo, the fate, the products of glycolysis, there are two: the ATP, the reduced NAD, and the pyruvic acid. Okay, and so the fate, depending on whether it's aerobic in presence of oxygen or in absence of oxygen. Now, in presence of oxygen, you will, the pyruvic acid, it is the same. We can also put it here. In presence of oxygen, they will be shunt into the mitochondrion where they oxidize it, or where they undergo further oxidation, produce energy. But in absence of oxygen, then they all undergo what we call fermentation. It could be lactic acid fermentation or alcoholic fermentation, depending on which organism, okay, to remove them from a cell, okay, into a form that can be accommodated by a cell. Okay? Yes. So now let's first talk about in presence of oxygen. The fate of the products of glycolysis in presence of oxygen. Hey, am I sharing the screen? I'm not sharing the screen. Oh, it's okay, guys. Sorry for that. If the challenge is severe, you can always unmute and speak. That's why you are not sharing the screen. It is okay. I'll come back and share. So let's share. So that's it. So now let's check on the fate of the products of glycolysis in presence of oxygen. Now, in presence of oxygen, of course, now glycolysis or respiration usually has three main stages. Okay, the glycolysis, the Krebs cycle, okay, and then the electron transport system, the ETS. However, there is this stage that they tend to ignore, but this stage is important. This stage is important. That without this stage, then there is no way how pyruvic acid is going to be fed into the Krebs cycle. So this stage is the pyruvic acid activation stage. That intermediate stage between glycolysis and Krebs cycle. It is equally important. And what happens? Our pyruvic acid undergoes what we call oxidative decarboxylation. Okay? And in the process, you have a complex protein. It's an enzyme, okay, that is found within the cell, and that enzyme is called the coenzyme A. Okay, so you undergo oxidative decarboxylation. You break down the pyruvic acid, okay, into the acetate. Acetate is just CH3, a diacetate, to an acetyl group. Oh, CH2. So this one, to an acetyl group. And now this acetyl group, okay, it is the one which combines with coenzyme A to form a complex called acetyl-CoA. So this reaction, this stage is important. Important. in respiration, like how I've said it. So pyruvic acid, which is a 3-carbon compound, undergoes what we call oxidative decarboxylation. Why is it oxidative? Because see, you are removing hydrogen from pyruvic acid. At the same time, you are removing proteins. Did I say proteins? You are removing electrons from pyruvic acid, and you are also removing hydrogen from pyruvic acid. Okay? So that is oxidation. That's an oxidation reaction. Now it is decarboxylation because you are removing a carboxyl group. Pyruvic acid, is it? I think it is CH3, CH2, okay? C, I think it is this. You can confirm, you can Google and confirm. So you remove a carboxyl group, okay? And at the same time, you lose these hydrogen ions together with the electrons. So the carboxyl group that is lost, it is turned into carbon dioxide, okay? Okay? And then the hydrogen ions that are lost together with the electrons that are lost in the process, they combine with the oxidized form of NAD to form the reduced NAD. Okay? And now together you form an acetyl group. An acetyl group. Now it is this acetyl group that will combine with coenzyme A, which is a complex protein that is found within the cytoplasm, to form acetyl-CoA. Now this enzyme, it is a transferase enzyme, where it is going to transfer this group, okay, from the cytoplasm into the Krebs cycle, into the matrix of the mitochondrion, where it will donate this acetyl group with OAA, oxaloacetic acid, okay, to combine together to form a 6-carbon compound citrate. And the Krebs cycle proceeds. Is that okay? Is that okay? Yes. Now, what happens with these metabolic poisons? Let me talk about them because you might find it somewhere. You might find it somewhere. Now, these metabolic poisons, what they do is they have some of them are non-competitive, of course, such as mercury, cyanide, and those heavy metals. They are non-competitive inhibitors. That what they do, they come and attach on this coenzyme A. Okay? They attach on this coenzyme A, and in the process, after they have attached, they inhibit this coenzyme A from attaching to the acetyl group. Okay? And if you inhibit this coenzyme A from attaching to the acetyl group, then you won't form the acetyl-CoA, and so there is no way how this acetyl is going to be shunned into the Krebs cycle. So in simple terms, you have blocked the entire process of respiration. So as a result, the organism will undergo just anaerobic respiration, which is not enough to produce the entire energy that is needed by that organism. So you won't meet the energy demand, and so the organism dies, okay, due to shortage of energy. Okay? Yes. So it is as simple as that. Now what antidotes do? Okay, let's also talk about antidotes, because you hear somebody has taken poison and then they have given him or her antidote. Now the antidotes for them, these are compounds that have a high affinity for that heavy metal than this enzyme. than this protein. They have a high affinity for the heavy metal than the enzyme itself. So as a result, when you take the antidote, then the antidote will remove, okay, the heavy metal from the enzyme and form a relatively stable complex between the metal antidote, okay, the complex is relatively stable, and you either pass out this complex in urine. It is excreted, it is removed out of your body by the kidney, and you pass out through urine. And somebody who was dying, now somebody regains the normal shape, the normal physical appearance, and somebody flourishes again. Okay? Yes. So that is pesticide chemistry. You can also go and find out more. But basically, that is this acid, this stage, the pyruvic acid activation stage. If this stage is not there, like I've told you, when you inhibit that stage, then there is no way, okay, the process of respiration cannot proceed. It must stop. A moment. Attend to your queries, to your concerns. Okay? That's pretty good. So that was that pyruvic acid activation stage. Now, after that stage, after the pyruvic acid activation stage, like I told you, okay, then you return pyruvic acid and you convert it into acetyl-CoA. That one is oxidative decarboxylation. Okay, you convert it into acetyl-CoA. Now I hope right now you know what acetyl is, what CoA is, coenzyme A. You know what it is. Now the acetyl-CoA, it is the one that will donate this acetyl group on oxaloacetic acid, OAA. Now OAA, it is a 4-carbon compound. Okay? When it receives when it receives that acetyl group, it becomes a 6-carbon compound. Okay? Citrate. So that's why in some books you find the citric acid cycle. This one. Why citric acid cycle? Because the first stable compound that is formed, it's a 6-carbon compound, and it is cyclic acid. Or in some book you might find a tricarboxylic acid cycle, or in others you may find Krebs cycle. This is someone's name. This is tricarboxylic acid because there are 3 carboxyl groups that are involved in the whole cycle and that are lost. Then finally you may find citric acid cycle, where it is the citrate, okay, it's citric acid which is a stable compound that is formed. So like I told you, it's a multi-step reaction in the mitochondrial matrix, during which the acetyl group, okay, is completely oxidized into carbon dioxide with generation of ATP in the cycle, okay? And it's one ATP molecule that is formed, but since there are two molecules running, so those will be two ATP molecules. And the ATP that is formed here is formed by the substrate level phosphorylation, and then the hydrogen ions that are produced, they are the ones that will reduce the NAD and the FAD. Now the NAD and the FAD, the reduced NAD is going to be transported to the electron transport system, okay? And the reduced FAD, it will also follow the same path, where it will be transported to the electron transport system for further oxidation to produce ATP by chemiosmosis. Okay? That's brilliant. So, it is named Krebs cycle after the formulator, the Hans Krebs. So this is someone's name. So this is the guy who studied Krebs cycle, and he's the first guy to enlighten us of Krebs cycle. Citric acid cycle, because the citric acid is the first compound to be formed, as we are going to see. And it is tricarboxylic acid because the citric acid formed is the first compound formed, and it has 3 carboxyl groups. You can imagine 3 carboxyl groups. And actually there are these 3 carboxyl groups that will be removed. Those are the 3 carbon dioxide molecules that will be lost in the process. Okay? As we are going to see. So, what happens? The 2 carbon acetyl group, like I showed you, it is CH3. The acetyl group is written as this, CH2. And it is bound to coenzyme Q. Sorry, coenzyme A, not Q. So this could be your enzyme. And the enzyme has some receptor site somewhere on the enzyme, okay, that will bind this acetyl group. So it is this enzyme that will now transport to form acetyl-CoA. Now it is this acetyl-CoA that will be transported, okay, within the matrix, that will now bind with oxaloacetic acid, OAA. Now 2 carbon, you are adding it to a 4 carbon compound, oxaloacetic acid, to form a 6 carbon compound, citric acid or citrate. Okay? Now the citrate that is formed, it also continues, okay, to undergo processes which will generate the production of energy. And the first thing that happens is isomerates, or it isomerizes. Now for the senior 5 today, I taught isomerism. You saw that it's where compounds having same molecular formula, they have different structure formula. So the citrate that is formed, or the citric acid that is formed, it isomerizes to form a more reactive compound, which is isocitric acid or isocitrate. Now the isocitrate is the one which undergoes oxidative decarboxylation. Oxidative, what does it mean? You lose the hydrogen, and the hydrogen is taken up by the NAD to form the reduced NAD together with electrons. Because see, this one is charged. You must move along with the electron, so that this NAD, okay, the NAD which is reduced, you can put a plus here, combines with hydrogen, okay, it's hydrogen together with the electrons to form now the reduced form of NAD, okay, NAD. Now this reaction is balanced because this electron will neutralize this positive charge. Oh, this is madness. So to form the reduced NAD, okay, and to balance the equation, you can put a 2 here. So there are 2 electrons that are moving. Why 2 electrons? Because the whole bond breaks. And the bond is made up of how many electrons? 2 electrons. But that is chemistry, that is organic chemistry, which we shall not go deep into. Now it is decarboxylation, because we have seen that we have 3 carboxyl groups. Okay? Now, of the 3 carboxyl groups, 1 is lost at this stage. Because you first ionize this, you remove a proton, okay? And then you remain with a carboxylate, okay? Or carboxylate ions, and then you decarboxylate, okay? Where you lose this as carbon dioxide. So that's why the whole reaction is called oxidative decarboxylation. Now, initially you have a 6C compound, citric acid, or a 6 carbon compound, citric acid. You are removing, or you are undergoing oxidative decarboxylation to form what we call, the compound that will be formed is called alpha ketoglutarate. Okay? Alpha ketoglutarate. Now, you think about this. Just keep it in mind. There is an amino acid called glutamic acid. Okay? Just keep it in mind. So alpha ketoglutarate. And this one, since from 6 you've lost 1 carbon, so this is a 5 carbon compound. Okay? It's a 5 carbon compound. Now, just let us erase this. Now our alpha carbon compound, or our 5 carbon compound, it does not stop there. It undergoes decarboxylation. Now in this case, it is still oxidative. So it is oxidized, okay, and it is decarboxylated. Still you are losing carbon, okay, carbon dioxide. Even here, you are also losing carbon dioxide. So it means from 6 to 5, now you've lost another one, so to form another 4 carbon compound, okay. So you lose carbon dioxide, you are decarboxylated, and you are oxidized. Now the compound that is formed, being unstable, it is attached on another coenzyme A. This is like a carrier protein, another coenzyme A, okay? And the compound, you're going to form a succinate, and the succinate will attach on the coenzyme A to form the succinyl CoA. Now the succinyl CoA, since it is a high energy compound, okay, since it is a high energy compound, as it is converting to succinate, okay, you have the enzyme called succinyl dehydrogenase. In the process, it is going to lose some energy, and now it is that energy that will add an inorganic phosphate on ADP to form what we call ATP. We are going to see, we have the illustrations down, okay, to form ATP. And the mode of formation of ATP here is called the phosphate level phosphorylation, because we are forming ATP from an energy that is lost from high energy compound. So now the 4 carbon succinate, it is now going to lose the 2 hydrogens to form, to FAD, to our flavin adenine dinucleotide, to form reduced FAD, okay, and for it turns into the fumarate, and then fumarate proceeds to marate, then fumarate is hydrated, and then it undergoes rearrangement to form marate, and finally marate is oxidized. Now this is not decarboxylated, you no longer lose any hydrogen, any carbon dioxide here, so it is just oxidized by losing the hydrogen ions to NAD, okay, to form how many? To form our reduced NAD, okay, and itself turns into oxaloacetic acid, OAA. Now, since it's a cycle, you must regenerate back. You remember in photosynthesis, you must regenerate back the RBOP, okay, the ribulose bisphosphate. So also here, you must regenerate back your oxaloacetic acid, okay, to complete a cycle. So basically this is what we've been talking about. Your pyruvic acid is activated to form acetyl-CoA. Then the acetyl-CoA donates the acetyl group, combines with citric acid. Then the CoA is given out, then to form the oxaloacetic acid, form citric acid, then isocitrate. Then isocitrate undergoes oxidative decarboxylation to form alpha ketoglutarate. Then alpha ketoglutarate releases the electrons to form what we call succinyl-CoA. Then succinyl-CoA, since it has high energy, then you release energy used to add the phosphate group to ADP. Then itself turns into fumarate. Then fumarate is hydrated. You add water molecule, then rearranges to form marate. Marate then loses electrons to NAD, okay, to form what we call oxaloacetic acid. However, this is just a summary. Let's talk about it in details. Okay? So this is our OAA, oxaloacetic acid. Okay? So it will gain an the acetyl group from acetyl-CoA, as you can see. Okay? And you have your coenzyme A here, and you add this acetyl group and you form your citric acid. Now this is the citric acid. However, you don't need to cram these structures. This is not biochemistry. You understand the structures when you go deep into biochemistry, but this is just biology, and we are just introducing you to this. Okay, so we shall form our citric acid. Now the citric acid, it will undergo what we call isomerism. So what is wrong with my PC? So it will undergo isomerism to form what we call isocitrate. It is just arrangement where there is loss and addition of water, just arrangement to form isocitrate. Then the isocitrate undergoes oxidative decarboxylation. Oxidative, you can see this is oxidation and this is decarboxylation. You are losing carbon dioxide in the process to form alpha ketoglutarate. Now this is a 5-carbon compound. Now the alpha ketoglutarate, it also undergoes oxidative decarboxylation, you can see, but in presence of coenzyme A, okay, to form what we call succinyl-CoA, and this one being a high energy containing compound, as it is releasing this succinyl to form succinate, energy is going to be lost, okay, and the energy is going to be received by the guanine diphosphate. It is GDP, it's here, okay, and when you add a phosphate group, you can see an inorganic phosphate within the cytoplasm, okay. So the inorganic phosphate, the energy that is lost in this process, okay, is going to add an inorganic phosphate to guanine diphosphate to form GTP, and this one being a high energy containing compound, okay, and it's unstable, it will dissociate there and then, okay, to release energy, and the energy now will be used to add an inorganic phosphate. Actually, the inorganic phosphate here, it will just be donated to ADP to form our ATP. And now this is the only ATP that will be formed in Krebs cycle. And since there are two molecules running, remember in glycolysis we split the glucose into dihydroxy acetone phosphate and phosphoglyceraldehyde 3-phosphate. So there are two molecules running, okay. So since you are forming one here, then plus the other one for the other molecule, so there are two ATP molecules that will be formed at this stage, okay. Then finally, the succinate that is formed, it will undergo oxidation, where it will release the hydrogen ions, and hydrogen ions will be taken up by this FAD, the flavin adenine dinucleotide, okay, to form fumarate, and then fumarate is hydrated to form marate, and finally marate is oxidized to form our oxaloacetic acid, and the cycle completes itself. Now, what are the products of NAD, of this cycle? Number one, you are forming ATP. And where is the ATP formed? It's here. Number two, you are forming 3 reduced NAD. Okay? 3 reduced NAD. NADH. One is here, another one is here, and another one is here. You need to know at which stage is where the NAD is formed. Then, number 4, you are forming 1 reduced FAD. FAD H2. And finally, carbon dioxide, maybe. Carbon dioxide. And how many molecules of carbon dioxide? Just 2. But since there are 2 cycles running per one single molecule of glucose, then we shall multiply everything by 2. Since there are 2 cycles running, then we shall multiply everything by 2. Is that okay? Okay. So let's check. Let me check the chat box. If there is any concerns and so on.
Yeah, where is the 2 coming from?
Where is the 2 coming from? I'm going to answer that. Who else? Another question? Another question? No question for me? Meanwhile. No question for somebody who asked where the 2 is coming from. You can see. It is in glycolysis. Remember, after forming that 1,6 fructose, okay, you can go back fructose or fructose 1,6 bisphosphate. After forming that compound, it's a high energy compound. It splits, okay, to give 2 interconvertible 3 carbon compounds. The glycerate 3 phosphate, or you can call it the phosphoglyceraldehyde, and then the dihydroxyacetone phosphate. There are 2 molecules that are forming here. Now the 2 molecules, when we followed, for us we followed 1. However, they are interconvertible. We followed this glyceraldehyde. Okay? But however, you can also convert this DAP, dihydroxyacetone phosphate, into the glyceraldehyde, and you feed it you proceed with glycolysis. But for us, we followed 1. So even here, we are still following 1. Okay? So if 1 is producing 3 NAD, reduced NAD, and 1 is producing 1 ATP molecule, and 1 is producing 2 carbon dioxide molecules, then it means that since there are 2 here that are formed, yet for us we are forming 1, then we shall just get everything, okay? We multiply it with 2 to get the total number of ATP molecules that are formed from 1 glucose molecule in Krebs cycle. The total number, okay, of NAD, reduced NAD, that are formed from 1 glucose molecule in Krebs cycle, and the total number of fat molecules or reduced fat that are formed from one glucose molecule in the Krebs cycle. I hope that one is okay. Okay? Yes. Any question, you can unmute and ask. It's okay. You can unmute and ask. Okay, let me check the chat box. It is still good, it is still fine. How is phosphorylation? How is phosphorylation done in terms of chemiosmosis? We are going to reach the electron transport system. We shall talk about chemiosmosis. Okay? We shall talk about that chemiosmosis. And I show you how it is done. But that was some pretty good question. That was a good question. So, you can compare the Krebs cycle and glycolysis. However, this is just simple work. Similarities, NADP is NAD, reduced NAD. Why NADP? NADP is found in photosynthesis. But this is just NAD when it is not phosphorylated. Okay? So this is NAD, reduced NAD is formed in both. ATP is generated in both. There is reduction in number of carbon atoms in both. And pyruvate participates in both. Where in one, the pyruvic acid becomes a product, that is glycolysis. But now in Krebs cycle, pyruvic acid now becomes a reactant. Then in both, they do occur in living cells. Then with differences, here you don't have their electron acceptor FAD, but you have their NAD. But here you have the electron acceptor FAD. Carbon dioxide does not form in glycolysis, nowhere. You didn't form any carbon dioxide. But now in the Krebs cycle, actually that's where carbon dioxide is formed. Okay? That's brilliant. Then occurs, you can also calculate the pyruvic acid activation stage. You're going to produce one carbon dioxide molecule, but since there are two running, then that's two. Then in the Krebs cycle, you form two, and since there are two running, that's four. So 4 plus, those are the 6 molecules of carbon dioxide that are formed, okay, during photosynthesis. And from that equation of yours you had in O-level and primary, I want you where you have glucose, which is 12, and oxygen, which is 6, plus oxygen. Are you seeing oxygen anywhere? Are you seeing oxygen anywhere? So do you see that this equation misleads? I'm not seeing oxygen anywhere. You form carbon dioxide, okay, and the carbon dioxide gas, and there are 6 molecules of carbon dioxide per glucose molecule plus water plus water, and still they are they 3? This is 6, so they, oh my god, so these are 6 molecules of water, and then we need the 6 here molecules of oxygen that are needed. So the 6 molecules of carbon dioxide, I hope you can account for them. Also, the 6 molecules of water, we shall account for them when we reach where water is formed. Okay? So you can account for everything as far as this process is concerned. Now this one occurs in the cytoplasm. The glycolysis, like I told you, it occurs in the cytoplasm, whereas Krebs cycle occurs in the matrix. And for it, glycolysis can occur in presence or absence of oxygen. But for Krebs cycle, for it to occur, then oxygen must be there and sufficient amounts. Okay? That's basically how simple it is. Now with electron transport system and chemiosmosis, we're going to talk about your chemiosmosis now. Okay? And you will appreciate. So the electron transport system, it is a sequence of oxidation reduction reaction, or you can call it a sequence of redox reactions, okay? Redox reactions, whereby the transfer of electrons between the protein complexes in the inner membrane of the mitochondrion. Okay? We are going to talk about this and you'll be able to appreciate. So the electrons are transferred between the protein molecules in the inner membrane of the mitochondrion is coupled with the transport of protons, of hydrogen ions into the intermembrane space, okay, to create a proton potential gradient, okay, that drives the synthesis of ATP by ATP synthase. If you understand that statement, then you understand what electron transport system is and what chemiosmosis is. However, we are going to talk about them in details. Okay? You have protein complexes within the inner membrane of the mitochondrion, and these protein complexes, they transport the electrons at a series of decreasing energy levels. In other words, from high energy level to low energy levels. And in the process, the electrons lose this energy, which the energy is used to pump the hydrogen ions into the intermembrane space of the mitochondrion to create a proton potential gradient. We are going to see what it is, which is going to be used to add an inorganic phosphate to ADP to form ATP. And actually, much of the ATP molecules, by the way, are produced within this electron transport system. Imagine 34 ATP molecules. Okay? 34 ATP molecules that will be produced by electron transport system. So the other ones, they are just like, which word should I use? They are just residues. Okay, they are not residues, but those ATPs that are formed in glycolysis, there are two of them, okay, and the two ATPs that are produced within the Krebs cycle, these four of them, they are just nothing, okay, but the ATP actually, the site of energy production is here, where you produce 34 ATP molecules, okay, and let's see how it is done by chemiosmosis. So for you, if the one who asked chemiosmosis, Jordan, here you are with your chemiosmosis. And the concept is just one. Chemiosmosis, you form ATP due to the proton potential gradient that is generated between the intermembrane space and the mitochondrial matrix. Okay? That proton potential gradient that is generated, you add an inorganic phosphate to ATP to ADP to form ATP. And how is it generated? You pump these protons in the intermembrane space. You actively pump. And where is the source of energy? It is these electrons which are moving at a series of decreasing energy levels, okay, within the protein complexes that are found where? In the mitochondrial membrane, in the inner mitochondrial membrane. So that is the concept. So the hydrogen ions, they will always move from a region where there is a high concentration to the region of low concentration. You can see the intermembrane space, this space, you have very many. You can even add protons, very many protons. So, but in the mitochondrial matrix, the protons are not that mob. So as a result, these protons always tend to cross. They always tend to cross. But remember the structure of the plasma membrane? They cannot just cross through. Okay? They can't just cross through. Why? Because of this phospholipid bilayer. The tails are hydrophobic. Okay? The tails are hydrophobic. And the heads are hydrophilic. However, the ions are positively charged. They are polar. So they can't cross this hydrophobic tail. So as a result, you need a protein complex, a protein channel within the membrane that will create a pore over which protons will diffuse through. Let it sink. Should I repeat? Let it sink. I said because the membrane is made up of a phospholipid bilayer with hydrophobic tails, like how you can see, these polar ions cannot just cross through the membrane. So as a result, you will need a protein channel that will create that pore over which these polar ions will diffuse through. Okay? Yes, first keep that. First keep that in your neurons. Now, the protein the proton gradient that is formed, okay, between these two is the one that is, okay, the proton gradient forms across the membrane that is not readily permeable to ions. I already talked about that. So therefore, the hydrogen ions or protons, they create an electrochemical gradient across the membrane which can harness the ATP production. I'm going to talk about that more. So you create, yes, somebody saying something. It's okay, you
So
can
what's
say.
the meaning of harness?
Pardon?
The meaning of harness.
Harness.
Meaning of harness. Harness.
Yeah, harness is an English word which means like helping. Okay? Like you help or you facilitate. Actually, yeah, you can use facilitate if you want to avoid help. So it is this proton potential gradient or the electrochemical gradient across the outside, so that the intermembrane space, this side and this side, that facilitates the ATP production. Now, within the membrane, you have there this protein molecule. It is both a channel protein, but at the same time, an enzyme. Should I repeat that? It is both a channel protein in that it's a transmembrane protein. Okay, you can see on this domain you have a pore, and it's this pore which will allow the hydrogen ions to freely diffuse through that pore. But on the other end, okay, on the other domain, it has a kinase activity. Okay, it is an enzyme, okay, that can add an inorganic phosphate to ADP to form ATP. Okay, that's brilliant. And there's something coming, have no worries. So this enzyme, okay, it is called an ATP synthetase or synthetase. You can see that makes ATP by chemiosmosis. It allows protons to pass through the membrane and uses the free energy difference to phosphorylate an ADP to ATP. That's not enough. Have no worries. I want you to assume this. If you have a stream of flowing water, okay, and you raise the water over high heights, for instance, okay, let me say this is, you're going to raise, this is a wall, okay, This is a wall, and you're going to raise water, push this water, pump this water until when it reaches this wall, and then you have something here that is going to rotate, maybe okay, a turbine. Actually, how electricity is generated, they build this wall to create that mechanical force, okay, so that they raise this water at high levels, okay, and then when it reaches this high level, then it will drop at a high speed with too much force, okay. Now this force, it is the one that is going to create these turbines to spin, okay. The turbines will spin. Why are they spinning? Because of the force that is generated by this stream of running water. Now as the turbines are spinning, I think they are connected somewhere that is able to convert this centripetal force that is generated within this spinning turbine into an electric energy, okay, or into an electric force, which can now be transmitted into generators, wires, into your wire, into your TV, and boom, you have electricity, okay. So this concept of this stream, okay, that creates that high centripetal force, this turning into the turbine that is converted into the electricity. It is the same concept that works here. Should I repeat? Let it sink. I said this force that is created by a running stream, okay, it is going to spin this turbine, okay, which will create a centripetal force, okay, by this turbine that will be converted into an electric energy. And then the electricity is now transmitted to your living room. You charge your phone, you attend my lesson. So that force is almost identical to this. Now let me wrap everything. Now, this enzyme, as you can see, it spins around. Are you seeing these arrows? They are not by mistake. You can see these arrows, they are not by mistake. They spin around, okay. Now since you have a very high concentration of hydrogen ions in the intermembrane space, okay, it creates a high electrochemical gradient, or you can say a proton potential gradient between the intermembrane space and the mitochondrial matrix. So now as these electrons are diffusing through, it's like falling of this water. It's like falling. You can imagine this mechanical force, this force that they will be having. So these protons, they also be like falling, okay, through this protein. Now as they are falling, they will be containing that potential energy, okay. Now it is the potential energy that will spin this protein molecule, okay, that will spin this protein molecule through. That energy, that potential energy that is lost by these protons diffusing through this ATP synthesis enzyme, it is the one that will spin this enzyme, this protein within the plasma membrane. Now as this protein is spinning, since it has the kinase activity on this domain, it will be able to generate enough energy that it will use to add this inorganic phosphate to ADP to form ATP, which is a high energy containing compound. And if it happens, you produce enough ATP, okay, and the ATP that is formed, it will be formed by chemiosmosis, okay. So this is the chemiosmotic theory of ATP production in the mitochondrion and 2 in the chloroplast. All these theories They are there. I hope that one is okay. Now, after understanding this, let's see how a membrane is organized. So anyway, I had a three catalytic sites in the stationary knob join the inorganic phosphate to ADP to make ATP. So basically I've talked about this within here. So you have that active site. As it is spinning, it generates enough energy that will bind an inorganic phosphate to ADP to form ATP. And boom, you have your energy. Is that okay? What is the time? It's 49 minutes. Can we proceed? Or you are tired? You want to first digest that?